Today we will discuss about T cell differentiation into Th1 and Th2 cells. In many of my immune cell videos, you have seen that T cells differentiate into Th1 cells and Th2 cells. So what are the functions of this Th1 and Th2 cells and how they are differentiated from T cells? We will discuss that thing in this video. So we will start with this. What are the properties of Th1 and Th2 cells? So Th1 and Th2 cells are differentiated from these naive T cells when they come and interact with the antigen presenting cells. And so naive T cells is usually represented as Th0 cells. So when the Th1 cells are formed, so they have the immune or tumor suppression properties or tumor regression properties, and they usually has this kind of receptors, that is interleukin 2, INF gamma and TNF beta. Whereas when Th2 immune cells are differentiated from Th0 cells, they has this tumor progression properties, and they usually has these kind of receptors, that is interleukin 4, interleukin 5, interleukin 6, interleukin 10 and interleukin 13. So how these Th0 cells differentiate into this Th1 cells and Th2 cells? What are the fate that determines whether Th0 cells will differentiate into Th1 cells or Th2 cells? Usually depend on many factors. So we will understand what are the characteristics from which we can identify whether the immune cells are Th1 cells or Th2 cells. So these are these THP cells. So THP cells, we can say that THP cells is the T cells, T helper cells which are polarized. That is, they can be differentiated either into the Th1 cells or the Th2 cells. And when they come and interact with this antigen presenting cells that has the information about the non-self epitope, these differentiations of this T helper polarized T helper cell polarized occurs. So during the differentiations, we will not go into detail. We will understand here what are the characteristics that are found in the Th1 cell and Th2 cells, how we can differentiate them into those different cells. So during these antigen presenting interactions, what will happen is that two kinds of information occurs. First one is to activate the T cells, and second interaction is to provide the information about the non-self epitope. And after that, the differentiation based on this information, the differentiation of this THP cells or T helper cells, T polarized helper cells occurs. And if those, so these are the steps where differentiation occurs. And here and after so here the interactions is occurs, and the T helper polarized cells is finally differentiated either into the Th1 cells or Th2 cells. So if they are differentiated into the Th1 cells, then they will have certain properties such as they have this IFN gamma receptors, and these IFN gamma receptors are found both in the mouse Th1 cells and also in the human Th1 cells, and this can be seen here. And if this T helper polarized cells is differentiated into the Th2 cells, then they will have the properties such as they have this interleukin 4 receptors, and however they do not have this IFN gamma receptors. This interleukin interleukin 4 receptors is slightly controversial as this slightly controversial in the human brains, not in the human cells, because this interleukin 4s are usually been measured on the heterogeneous populations that contains both the Th1 cells and Th2 cells. So because of this, right now there is not a consensus that Th2 cells has human Th2 cells has this interleukin 4 receptors. So if the T cells has this interleukin 4 receptors, then they are more prone towards Th2 cells, and if they have this IF and gamma receptors, they are more prone towards Th1 cells. And other things, when the cells are under this THP stage, they do not have CD8 plus positive. However, when they differentiate into this Th1 and Th2 cells, they will have CD8 plus positive receptors also. And at this stage, when the cells are that this THP stage, they are called as this progenitor T helper cells, polarized T helper cells. That is, they are ready to differentiate into these Th1 cells and Th2 cells. So I hope you got a basic idea how to characterize Th1 cells and Th2 cells, and which of them is beneficial for our immune system, and which one of them is beneficial for the tumor progression. If you need to understand more about this topic in detail, you can go through the references that has been provided in the description and also in the bottom of each figures. That's all for this video. Thank you for your time.